Okay. The room suddenly looks a bit bigger when you're sitting up the front. Seems like there's more people. Right. Cool. I'll start with a few slides. So I'm going to be talking about a tool that I've developed as part of my PhD / part of the SCRI Tools for Polyploids program. This is called MCHap. It's a tool for microhaplotype assembly, and we'll get into a bit more about what that means shortly. I think just sort of to give an idea of the scope of the tool, where Chris just presented a workflow joining up a whole lot of tools to make a sort of complete and whole workflow. MCHap is the kind of tool that would make one step in that workflow. So this is not an end-to-end analysis. This is expecting you have some inputs ready that have come out of a previous analysis, and it also expects that you're doing some downstream analysis afterwards, although it doesn't prescribe what that should be. So it's just one step in a larger pipeline. It's a command line utility, so it's written in Python, but it's designed to run from a bash prompt or something like that. It's essentially a variant caller of sorts. However, it requires variances input as well and produces more complex variances output. So the tool itself is actually composed of several smaller tools. The main two that we use and we'll talk about are MCHap assemble and MCHap call. So they have quite similar interfaces. MCHap assemble is used for de novo assembly of haplotypes, so where you have SNPs that you're interested in assembling into haplotypes. MCHap call is used for recalling genotypes from a set of known haplotypes. And we'll see sort of the difference when we go through them. But hopefully if we get far enough through this, I'll get to what I refer to as the two-step approach, where you actually use MCHap assemble followed by MCHap call on the same data set, and there are reasons why that can give you better results, particularly when you're working in a set of related samples. So just some background on the data that we're going to use. This is a very small subset of capture GBS data. So this is targeted sequencing, targeting some exons across the genome. And we get this kind of pyramidal or bell-shaped read distribution. So we typically with this data, we just assemble sort of the central part of the data. We don't want to have too low a read depth for haplotype assembly, particularly in polyploids. So MCHap was originally designed with this sort of data in mind. However, it can work on any sort of NGS data. So if you have whole genome, you can define small windows across the genome to assemble. It will start to run into problems if you have lower and lower read depth, however. So MCHap, rather than being a sort of statistical mapping tool to create haplotypes via segregation ratios, it tries to assemble haplotypes in each individual sample individually. And to do that, it exploits reads that have overlapping, reads that overlap multiple SNPs or multiple SNPs. So if you have if you have a boring organism that only has the occasional SNP, and so you don't get much overlap of SNPs on a single read, then it might struggle. So we need to have NGS data where you have overlapping multiple SNPs like this, which is typically the case in a lot of the polyploid plants that people here work in. So to turn these reads into haplotypes, it uses a Markov chain Monte Carlo approach. This is a Bayesian tool. I won't go into the full details of this, but essentially the MCMC is starting with a random genotype, seeing if that it could be a randomly proposed genotype. We try to be a little bit smart in the way that we started the algorithm. And then it runs through a series of steps. In each step, it updates that genotype based on a ratio that's calculated from a likelihood function. And the idea is that it's iteratively moving and improving that genotype till it gets a more and more realistic estimate. And we try and do this relatively fast. It's fast for an MCMC program, but it's still probably slow compared to other variant callers. And we do this for a large number of iterations, and then we look at the proportion of each genotype in that chain that we've created of different genotypes, and essentially the proportion the proportion of time that the algorithm spends in a given genotype is an estimate of the probability of that genotype. So MCHAP doesn't just produce a best call, it produces probabilities around those calls as well. So you've got some quality control data. Cool. Okay, I think we'll jump into it. So I'm just, this is just on the same server you've all just used. We're also going to be using Docker here. However, we're not going to be using it properly because just put MCHAP into a Docker container to make it consistent, so we're all using the same version. So in this case, we've actually installed the software is installed in a Docker container, but the example data we're going to use is also inside the Docker container. So we're going to actually run the Docker container and then log into the container at the same time. So I'll start typing that command. And I'll paste this into the chat. Cool. So hopefully you can all use that command and run it. Sorry, this needs to be running a camera.
Yeah.
So once this is running properly, you should see that you're the root user inside the Docker container. If I type ls here to list what's in this directory, you'll see that we're in what looks like the partition of a Linux installation inside the Docker container. And you'll see the first folder is MCHAP, which is, we'll move. So CD to change directory.
That's the keyboard different.
Yeah.
I think this is keyboard.
Yeah.
European.
Yeah, I was like looking for the one.
That would be confusing.
Yeah, I was
That change.
Interesting.
My hero
The Z is there, the Y is there. Maybe the frequency of keys.
So now we're listing.
I'll also post in a link to the GitHub repository, so all the documentation is on here, and the example we're roughly going to be following is the example that is linked to here in an example notebook. So we won't be doing it exactly the same as in this notebook, but if you want to refer to something later, similar sort of information.
Okay, so what you posted in the chat. Okay,
Oh, this is looking at this one.
so he's going into. So he just posted in the chat kind of roughly what he's going to show us today. But you don't need to. Yeah, it doesn't matter. Like, yeah, you don't need it. It's just like if you want to go back to refer to it. So he's done this example. We just navigate to examples. So the input directory is CD.
So just wait to see the story.
Yeah, there's no chat yet. We can't log into his chat.
Oh, it's not logged in. Right, okay, yeah.
Oh, okay. Hopefully not too much typing.
So essentially.
So if you're waiting, you can type just on its own and run that, and that will print out the list of all the subprograms.
This is a list, and this is a type that you can which I assume is going to be loaded in Oh, you have to share it. Next to one. So this is the.
Yeah, so that's.
So if we look at the options here, we can see 4 different options. Assemble, which is the first tool we use, then call, which we'll use later. There's also a third program called exact, which does a very similar thing to call, except doesn't use an MCC, it uses a brute force approach. That's not recommended, it's really using for testing and comparison, but it is an option there if you, there are some situations where that can be a faster approach, but generally we don't use that. The final one is version, which just prints the version. So to run a sub-program, type mchap, then version, and the version program will just print the version of the tool.
So what he's now done is just show version of the tool. And he's got the same version as
So I'm going to type clear just to bring up some more space on the screen. I hope can is there someone got sound on? So we'll just have a look at the example data. So use ls to list all the input, all the files available. So we have an IPython notebook there called biparental. That's the actual example that's listed on the website. We're not going to use that file today. Feel free to delete it if you want. But in the input, we'll see more subdirectories. So we've divided our input by file type. So in input BAM. So these are our BAM files that contain our aligned sequences for all of our samples. So this is a step that you need to do prior to running mchap. So in a workflow like reads to map, it'll have an alignment step and so on. So these are BAM alignment files aligned to a reference genome. In this case, this is a very small subset from a biparental cross. So we have 2 parental samples, parent 1 and parent 2, and then we have 20 progeny samples, and we've just got a single BAM file per sample here. mchap can actually work if you have multiple samples in a BAM file, in a single BAM file, but it's generally more efficient to have 1 sample per BAM file, and I find it way it's BAM. Cool. So if we look at input, so mchap, particularly running the assemble tool, requires 4 inputs by default. The first of these is the BAM files that contain your sample alignments. We also require a bed file, which defines the windows that we're going to assemble haplotypes in. So if we have a look in bed, or even we can see there's 2 files in there. Targets 4 is just a subset of targets 20, and we'll use the smaller subset today. So if you're not familiar, you can use less to read the file. So if you have a look inside that bed file using less, then you can see This is a very small bed file defining 4 different windows all on chromosome 1. So this is first column is the chromosome name, then the start position of the window, then the stop position of the window. These are all about 120 base pairs. And then the 4th column is optional. It's just a unique identifier for each locus. It's not actually needed, but if you do have it, it's quite handy because that will be reproduced in the output VCF file, so it makes it easier to link up the data. So press Q to exit from less. And in input faster, we just have our reference genome, which in this case is just chromosome 1 from Red 5 version 2. I can't remember the exact genome I'm using here, but it's this is actinidia, so kiwi fruit data, tetraploid kiwi fruit data. So this is just chromosome 1 of the genome. And in input VCF, we'll see a few files. So this is a VCF file that defines our input variants that we're going to use. There's an uncompressed version, a compressed version, and then an index for that compressed version. So if we have a look inside this VCF file, we can see that this is a very, very simple VCF file. In fact, I just wrote a script to produce this one for me. This VCF file defines the SNPs that our assembly is going to use to assemble haplotypes. If you have variation in your data set that is not in this VCF file, then it will be completely ignored. So you can reuse a VCF file from anything for as long as it's using the same reference genome, but it makes sense to produce a VCF file that is relevant to your current data set that you're working on. Typically, I would do some initial just SNP calling using something like GATK or Freebase, produce a VCF of SNPs that way, and then use those to assemble the data set. So you're kind of doing multiple phases of your encoding. One to get your SNPs, the second one to assemble those SNPs into variants, into haplotypes. One thing, if you're familiar with VCF files, you notice here is we don't actually have any sample data in this file. That's just to make the point that this input VCF, it's not using any of the sample data for it, it's just using it to define those variants. If we scroll down a bit here, you'll see we do have some triallelic variants in here as well. MCHAP can only use SNPs, but it can use multi-allelic SNPs. So if you have triallelic or even tetraallelic SNPs, it can make use of them. Cool. So to run MCHAP assemble, we use MCHAP assemble. If you run that on its own, it will print out a lot of help text. There are a lot of parameters that can be configured for this tool, which can be a bit overwhelming. In practice, you generally need to use only 4 or 5 of them or something like that. So we'll work through the basics that you should be aware of. And then I'll put quite a bit of effort in trying to make sensible defaults for most of the parameters. So it should work just out of the box in most cases.
So he didn't see that.
Clear. Assemble. To input our BAM files, we type --BAM and then the path to those files. And in this case, I'm going to use a star, so a wildcard, a star, which just means use every BAM that's in that folder. It will match up to every BAM file available in that folder.
The star means that's the wildcard.
We need to tell MCHAP the locations that we want to, the windows that we want to assemble into haplotypes. So we use the targets parameter for that, and that's what bed. And we'll use targets 4.bed. It will work with targets 20.bed. It'll just take a lot longer, and there's a lot of us, so it might not be a good idea.
So what's the character after BAM?
After BAM?
Yeah, I can't see.
Oh, yeah, sorry, that's the cursor, yeah.
I was like, that's strange. There's a character there.
I will post this into the chat, those that have the chat.
So the targets do that.
Oh, yeah.
Yeah.
Awesome, thank you.
We just, so essentially this tool comprises BAM files and CF files. So we're just telling them That's all that we're calling. Are we telling where the band
Um,
files are?
In this one, make sure you use the .gz, which is the compressed version of it. And the final one is so maybe I'll just run this first by myself to check that I've got the spell everything correctly.
Separating commands.
Yeah.
So this is running now. The way it works is it prints out variants as each locus as it's been assembled. So it's initially it's creating the header of a VCF file, so we can see it contains the VCF format, the date the file was created, the source of the file, so the version of MCHap. It actually copies the entire command line argument we use to run the program. In this case, it's expanding that wildcard to the list of band files. And then there's a whole bunch of header information followed by our list of our samples. So this is running in the background now, and once it assembles each locus, we should see the results of that come up as well. Now, MCHap uses a compiler called Number to compile some of the core parts of the Python code. Usually this will compile the first time you run the tool on a new computer. So what that means is the very first time you run the tool on a new computer, it might run slower than normal. But each time when it runs, the compiled functions are cached, so they should be available for future runs. So it's just it's a bit inconvenient, but it can confuse people when things take a very long time, even for a small example, the first time you run it, but it should be faster following runs. So by default, MCHap will just print the results to the terminal. It will print the entire VCF. This isn't actually very helpful, so we'll show you what to do with that in a second. But the first thing you might notice, maybe not the first thing, there's a lot of text there, but we can see that in VCF format, all of these individuals have been called as diploid, even though they're tetraploid. So you need to specify the ploidy of your organisms, and it will default to diploid if you don't say anything. So if I type clear, and then I'm going to use the up key twice just to bring up the command that I previously typed. So one more argument to add is ploidy 4. Just in the interest of time, I won't run this just yet. I'll also put in inbreeding 0.01. So these are both optional parameters, but they're good ones to include. Ploidy is pretty obvious. If you don't put in the right ploidy, you won't get the right ploidy if variant calls out the other end. The inbreeding is just an estimate of the expected inbreeding coefficient. Now, you don't have to be particularly accurate with this. The more accurate, the better. But it will default to 0, which expects a fully outcrossed individual. And that is a bit of a problem, particularly in breeding programs. It's quite common for stuff to be at least slightly inbred, particularly with auto polyploids. You have things like double reduction that do actually contribute a bit to the inbreeding coefficient. But more importantly, MCHap assemble by default is assuming that your samples come from a population in which every possible haplotype exists, which is an unrealistic assumption, but it's kind of the only assumption we can make because we don't know what haplotypes are in the population in the first place. What that means is if you don't have an inbreeding estimate in there, it's quite likely that your genotype calls are going to be excessively heterozygous because you're saying they come from this incredibly highly heterozygous population. Setting an inbreeding coefficient, even just a small one, but like 0.01, means that it makes it easier for MCHap to call more homozygous calls because it's saying, okay, they might come from this unrealistically heterozygous population, but there is a chance that they have double copies of a single allele or even triple copies of a single allele. So this is something I might actually change the default in future. Default of 0.0 seemed reasonable at the time because people usually don't expect or often just assume things aren't inbred, but I actually think it's more realistic to have a small inbreeding coefficient. So even if you know nothing about your population, I'd say it's still better to estimate a small inbreeding coefficient. So if we hit run now, that would run again, except produce tetraploid calls. It will be slightly slower calling higher ploidy organisms. But rather than just printing it out to the screen, what we're going to do is use the pipe symbol, which is Shift \/ on a US style keyboard. So we're going to redirect the output of the program into another program, which is called bgzip. So bgzip is a part of HTS Lab, so a very well-respected bioinformatics library that's used to compress various file formats, including VCF. It compresses it in a way that makes it allows us to create an index for that file later, which means we can have faster access to it.
So he's just zipping it up, so it's making compressing the file.
Okay, so that final one is the name of the file that we're going to create. So we're running mchap, specifying our parameters, piping the result into bgzip to compress it, and then using that arrow symbol to direct it into a file, so we save it as a file. And I'll hit run there, and it could take a little bit of time. So hopefully it should be about a minute or something at most.
I didn't do the recording.
Interesting.
I can stop this on the back.
So controls.
He didn't put where you.
Then
Just go back.
Don't know why it's doing that. Is she using the example data?
She's probably not wrong directory.
Yeah.
Oh, yeah, she's probably running not as
Yeah.
Zipping it up so we remember.
Okay. Yeah.
But this makes you know what you
And so it's this one up here. So
Don't forget that one.
Yeah, right. Okay, she might be in the wrong directory.
it might be that, or it might be that she's pointed it to a different directory other than the faster one.
Yeah.
It's also possible that somehow in version it. I said it's not indexed, not that it's out of date. Sometimes
Yeah.
touch a file that makes the index look like
Yeah, because we can't, it's not.
Anybody do that? Make
Okay,
copy with kill.
so once you see your prompt come up again, it should have run successfully. So if I type ls, we can see, oh cool.
It should be control.
We can see that we have assemble.vcf.gz. So that's the file that we've created.
So we're only going to get the return once
And
done.
just out of good practice, we'll create an index for that now using Tabix, which is again part of HTS Lab. This is standard bioinformatics software. So, okay, so that command is saying we want to use Tabix to create an index for this file, and the pvcf is telling it to use the VCF profile, is the word they use. But basically, we're saying index this, it's a VCF.
So is that the comment?
Sorry?
I can copy that in.
Yeah. And after that, we'll see we have assemble.vcf.gz as well as assemble.vcf.gz.tbi. Now, if we want to have a look inside that file, I think Les will understand the. So we use Les to look inside these files. Les, the name of the assembled file, and now we can have a look through this. So all of these lines that start with a hash are part of the header information in a VCF file. So they include a bunch of metadata about how the file was created at the top there. Down further, we have descriptions of the various fields that we, or records we have for each sample. So if we look here, we can see the list of all of our files. So this is the order that they've come in. This should be the same order that the BAM files were entered in. Check. And then we get down to our first variant, which starts here. So this is a standard VCF file. However, the main difference is that rather than having small variants at each locus, you've got these much larger variants. In this case, we've got 120 base pairs. So the first one here is the reference allele. That is just whatever the reference genome is there. Even if it hasn't been identified in any samples, it'll be recorded from the reference file that we used. Following that, we have our alternative alleles. some of this metadata here includes the count, so we have 3 alternate alleles here. If we look at the SNV pos down here, this is positions of the single nucleotide variants within the reference, within the haplotype region. So we can see that in our VCF file, we had that's 1, 2, 3, 4, about 10 SNVs that fell within that window that we assembled. So each of those SNVs should have been included into these haplotypes. If one of those variants isn't present, so it's just the reference allele, it means it didn't find sufficient evidence of that alternate allele for the SNV to include it in any of the haplotypes. And this is one of the advantages of using a haplotyping approach, is it can actually reduce the number of variants you have quite substantially, depending on how closely related things are to one another and how variable they are. So each of our genotype calls is a tetraploid call. I will say that there is a way that if you have a mixed ploidy population, you can actually specify the ploidy of each individual using an additional file. And again, with the inbreeding estimates, if you have more specific inbreeding estimates for your population, you can include an inbreeding coefficient per sample, so you don't have. I'm not going to go through every field here because VCF is quite big and verbose, but these are. Is there anybody here who's quite unfamiliar with VCF format, or are people reasonably comfortable with it? Nobody said anything, so I'm going to assume you're all comfortable.
Depends what you mean.
Yeah.
Yeah, I know that it's got like, a header with information.
Yeah, gotcha. Yeah. So this is the genotype call VCF file. Let's just. So for the first individual, which in this case is the first parent, it's been called with a genotype that has 3 copies of the reference allele, which is the 0, and 1 copy of the first alternate allele, which is the 1. So that means the actual genotype for this has 3 copies of this allele up here, and it has, and 1 copy of this first alternate allele. So they use these numbers in the genotype calls to avoid having to print out the full allele every time.
So, Tim, also, so each row is one haplotype?
Yeah, so it's wrapping around here because I don't have much room on my screen, but all of that there is a single line in the VCF file. So that's one single line that's just wrapping over because I don't have enough room on my screen. And so the variant starts with the location of it. So chromosome 1, the start position of the record. And in this case, we have the locus name because we had that in our bed file at the start of it. So that's the advantage of having that optional locus name in the bed file, is it will be included.
So is this the location there in the genome?
Yeah, yeah. So that's the start position of the genome, of the variant in the genome. Down here, we also record the end position, just for convenience. So in a VCF file, the start is compulsory, but the end is optional, which is why it's recorded in a different place. And then these SNV positions, these all, to work out the position of a SNV, it's the start of the haplotype locus plus the position of the SNV. It might be plus minus 1 because it uses 1 base indexing in VCF, which is standard, which confuses me. But yes, you'll be able to work it out. And then for each of our, this next column, so we talk about columns in VCF that are separated by tabs. That column that I've highlighted there, that's the information for this particular record. So that's capturing metadata about the variant locus as a whole. And then that's followed by the sample data. So this first, the final column before the sample data is a description of the fields that are in the sample data columns. And then the first sample, so the first parent, this is the data for that specific for that parent. So it starts with the genotype, then it has various quality scores and so on. Actually, the one for the second parent is a bit more interesting because we can see that these last metadata fields I might need a bit of a better description of that, Elena? It's not based purely on the counts or observations. It's got a probabilistic function in there, so it's not that it requires this many observations of that read to call the allele. It takes the numbers of observations of an allele, and it also takes into account the inbreeding coefficient, the ploidy, and then it produces probabilities. So these probabilities are output for the genotype call are output in the VCF file. So this GPM here is the genotype posterior mode. So that's the probability of the genotype that's being called. And that's this 0.48 here. So for this parent, it's produced a genotype call, but it's got, it's actually a less, saying there's a less than 50% probability that this is the correct call. So we try and produce as many calls as we can. However, we're not guaranteeing, we also include metadata about how certain we are of that call. And so in this case, it's not actually that certain. The second value here, so PHPM it's very similar and it'll often be the same value, but it might be slightly higher. And that's a probability that at least the unique alleles in that call we think are correct. So the first probability takes into account the dosage, where the second probability doesn't take into account the dosage. If we scroll down to the next variant, we can see, first of all, we've got a lot more haplotypes, which is suspicious. Often when things go wrong in MC-Hap, it results in a more heterozygous call rather than a less heterozygous call, in fact, almost always. And we can see down here, so if we look at, so the first parent here has a probability of 0.8, so not particularly high. The second parent has a probability of 0.08, so that's less than 10% probability, so that's a very unreliable call. You'll also see here for the alleles called in here, the fourth one is a dot, which means that particular haplotype didn't reach the threshold to even be reported by the program. So MC-Hap doesn't report every haplotype that it looks at by default, because that can end up being literally millions, or for a large enough loci, even billions. And so it just creates a very bloated VCF file. So it has a rule which there's a threshold that you can configure, and by default it's 0.2, so 20%. And what that means is MC-Hap assemble will only report a haplotype if there's at least a 20% probability of it occurring in one or more samples. So that's a configurable threshold. 0.2 is a reasonable value for most cases. If you really want to get out all the fine detail, you can turn that all the way down to 0, which I wouldn't recommend. I'd turn it down to 0.001 or something like that. If you really just want only the really reliable calls, you could turn that up to 0.8 or something like that. And so this is the results we've got here for this for this assembly are not great, and that's because we're using because of that prior I mentioned earlier, that it's assuming the samples come from a population where every possible haplotype exists in an equal amount, which is an incredibly unrealistic assumption, but it's the only thing we really can do when we know nothing about the sample. So I'm just going to switch back to my slides here. So this is kind of a representation. This is kind of a representation of what that prior looks like. So you imagine you've got a list of all the possible haplotypes that can occur there, and it's saying they're all equally likely. What we want to do is have a more informative prior like this, where we say, okay, we're calling haplotypes in this population. However, we know that out of all of the possible haplotypes, these are the only ones that we expect to see in the population. Or an even better approach would be have specific probabilities around each of those haplotypes. And that's where we can use this two-step approach to run MC-Hap call on the output of MC-Hap assemble. We are getting 10 minutes away from lunch, so maybe I might have to stop explaining. What I might do is just jump onto running MC-Hap calls so you can at least see how the second tool works. So the first argument is exactly the same, which is the location of the BAM files. Now, instead of having a variance argument where we specify the SNP variance, we're going to use a haplotypes argument, and that's going to be specifying a VCF that already contains haplotypes. And so for that, we're actually going to use the file that we just created as the haplotypes file. So that assembly file already has haplotypes in all of the records, and we're going to reuse those haplotypes. So those are the only two arguments that are required for MC-Hap call, but we'll still include haploidy and our inbreeding. Yeah, we'll do that as well. So use a pipe symbol into BGZip. Read it to okay. I'm going to post that. I'll run that. This call tool will generally run faster than the assemble tool. There can be occasions where it doesn't, and that's usually because the VCF with haplotypes in it has just a huge, enormous number of haplotypes, and it's having to check every one of them. So. Cool, let's run. So the idea here is that with this two-step approach, the idea is that we're using MC-Hap assemble rather than caring about the individual genotype calls we're using it just to identify the haplotypes that are likely to occur in the population. So we're essentially using MC-Hap assemble to try and identify a more realistic prior for our population as a whole. The reason we think that's a realistic thing to do is because this is a biparental cross. All of the individuals are very closely related. So if you find a haplotype in one of the individuals, that haplotype should occur in other individuals as well. So essentially, if we have some bad samples in here with lower read depth where we're not getting good haplotype calls out of them, we're using the haplotype calls from the other individuals to improve the calls for that individual. This is still a bit unrealistic in this example because for those haplotypes that we have identified in the population, we're then saying that they're all equally likely to occur when we rerun it. There is more advanced options we can use to actually use the frequency of those calls from the original assembly and the downstream calling, which we won't have time to do today, but a lot of this two-step approach is about trying to refine a more realistic prior distribution or a prior probability for your haplotypes.
And to kill you for control C.
So I won't bother to index the second file because we're not going to use it in anything else. But if we use less call, you can see it's similar to VCF to before. We will see the exact same haplotypes listed here as we saw before because MC-Hap call uses the exact same haplotypes that were in the input file that we put into it, and so it reports all of the haplotypes again. With MC-Hap call, there is no posterior haplotype threshold option. So every haplotype that was input will be reported, which means with MC-Hap call, you'll never get a genotype call that has a missing allele. They will always be complete, which is one of the nice effects of the two-step approach as well. And if we look at the probabilities, we'll see so where are we? So this is the second parent here at the first allele, and we can see that is it? Yes, it is. we can see that the posterior probability here for that second parent has gone up to 0.8, where before it was less than 0.5. And it seems kind of ridiculous that, oh, we'll just run it through again and now we get better probabilities out. It seems like bullshit. But the reason is that this is a Bayesian approach, and so changing the prior distribution changes the posterior distribution. So by restricting the analysis to only look at a subset of the possible haplotypes, the posterior probabilities for the genotype calls will be higher. And so it is a different probability, but it's now a probability that's conditioned on the assumption that all of the haplotypes we put into this program are the only haplotypes present in the population. So as long as your prior is realistic, then it is a realistic improvement in the posterior probability. If you start having problems with your prior, then that's when you get weird results out the end that don't make sense, but will make themselves look more confident than they should be.
Is there a sensible way to get a good prior?
I think the two-step approach is good so long as you're dealing with a population of related individuals. That doesn't have to be a biparental cross. It can be a more complicated population. There are more sensible ways that currently aren't implemented that you could get priors as well, but that gets more complicated. Ideally, you would actually have a prior per individual rather than a prior for the whole population, but a population like this is okay. I have used MC-Hap on populations with much more complicated pedigrees where you have, say, you're including a founder that has only been used once in a very large population. That means that that founder is likely to include some alleles that aren't seen in any other samples because only half of its alleles will be inherited. So if you use this approach here, you're probably going to get a biased genotype call for that founder because it's very likely to include alleles that are rare in the population as a whole. So you do need to think about population structure when doing this approach. I'm working on sort of, particularly for that use case, I'm working on an approach that can incorporate more pedigree information so that you can have a strong prior where you can, but you also allow those founders to have their haplotypes called largely independently or just using information from the few progeny they have.
So what will you do when a parent is not known, when you have only one parent and the other is unknown?
So in a biparental cross, as long as you're not expecting an incredibly strong selection, you could still use this approach because as long as a reasonable size population, I mean, 20 is probably about as small as I'd go for this assumption, but this is actually a subset of a population of around 200. In a biparental cross like that, if one of the parents is missing, you would still expect all of its alleles to be found in the population as a whole. And that's where I'm actually hoping the pedigree-based approach would actually potentially allow you to impute that parental genotype as well. I mean, you could impute that as a downstream step by, so a lot of this is about trying to make reasonable assumptions about your population. The main problem we're dealing with here is that when you start creating haplotype alleles, you have so many possible haplotypes and so you're trying to refine that sort of parameter space down to as fewer haplotypes as is reasonable. The maths behind it for biallelic SNPs is quite simple. If you have 10 SNPs in a haplotype window, then you have 2 to the power of 10 possible haplotypes. And so I think that's 1,024 or something, I can't remember. But it's exponential complexity, and so it just gets absolutely massive very quickly.
So you have to exclude all outcrosses before and identify them before and then?
Sorry?
So if you have outcrosses in your population, that can happen.
Yes, yep. So you don't have to, but if you don't want to analyze them downstream, you'll probably get a better result if you do. So if you say you had a single outcross in there, so with a missed paternity and unknown father, then if you had a single outcross in there and used the two-step approach, then you would probably bias that individual towards looking more like the rest of the population, which might mean that you actually fail to detect it. In the past with this, I do a lot of sort of QC around my samples looking correct based on either the SNP calling data that I originally generate or the initial assembly results. So even though MC-Hap assemble produces less confident results that can be noisier, for sort of kinship-based, trying to identify outliers, it still actually can be fairly useful for that. So you do have the option of just using the MCHAP assemble results directly, but they will be noisier. If you have really high read depth and really quality data, though, that may actually be enough because it's a Bayesian approach. The more information you have or the more information you've put into it, the less effect the prior distribution has on the posterior distribution. There are arguments in here that you can use to put out more information about the posterior distribution. So I won't get you guys to run this in the interest of time, but if I type so this is using the report option, which we can use to report additional metadata, so not the default data. And in this case, it's the AFP field, which is the posterior allele frequencies. And so this will report not just the genotype call for each individual, which is the posterior mode, but it will report the posterior allele frequencies for each allele in each individual, which is a more sort of, you can think about it as another way of genotyping, but rather than saying it has to have exactly this number of alleles, you can say, well, it's got this probability for each allele. And this is actually what we can use to refine that higher for the second step more. I don't know if I should have run that, I might take a look. It's lunch now. I might just quickly jump to something that's not on the agenda. But I was I'm just aware that dealing with raw VCF like that is very difficult. It's not a nice way to deal with data. So I wanted to show another tool that I'm involved with, actually outside of the SCRI, but I'm hoping to have better linkage there in future. I'll try and present on it at the next major meeting, I think. But this is, yeah. So this is a Python library called SGKit, which is used for, it's quite a generic statistical genetics toolkit. So most of the developers on it work on human stuff, so they're only interested in diploids, but they've been quite accommodating for allowing me to put polyploid stuff into their toolkit. So you won't be able to run this. This is not installed on the server at all. This is just running locally on my machine. In this case, I'm running on the assembly results that we just produced, except this is for the 20 bait targets rather than just 4. So SGKit has its own, it uses a format called ZAR to store the data rather than VCF, so we convert the VCF to a ZAR file. ZAR is a generic multi-dimensional array storage format, which is good for HPC. Another whole topic I'm not going to get into. But once we load that, it represents our data as a bunch of separate arrays. And so this is showing the shape of the array in SGKit, so we have quite nice documentation available. Everything is treated as an array of data. In this case, the genotypes array is treated as a 3-dimensional array where you have variance times samples times ploidy. So every individual allele call in an individual is somewhere in that array. So it's a 3-dimensional array. We also have a lot of other information from the VCF here, and you can read in more as well. So that's that same array. If we type values or compute, so by default, SGKit doesn't read in the data. It uses lazy evaluation models. So you're basically planning out a compute pipeline, and then you hit compute at the end to actually run the analysis. So if we run that, this is just telling it to read in the genotypes and display them to us. So they're represented there using that same numbering system used in VCF, except the full stops are placed with -1. Because full stop isn't in it. There is some fancy functionality here that we can use to display the genotypes more like you'd see in a VCF, which can be quite nice. That's really as far as I was going to take it with this. I'm happy to talk to people more about it, but basically there's a limited number of tools out there that are generic for polyploid or multi-allelic data, and this is one that's fairly new. We're hoping to publish this year, but it can support both polyploid and multi-allelic data in one package. Right, I think it's lunchtime, unless anybody has any questions. My throat's giving out. I've done more talking this week than I've done in the last 6 months.
Do you have more to do after lunch though, Tim?
No, we're good.
Okay, so we're on track.
Yeah.
All right,